Good afternoon, everyone, and welcome to the 2021 Tyson Summer Seminar Series, virtual edition number 2. Hopefully next summer we'll be in person again. We are very fortunate to have a really good lineup of speakers this year. And before our first speaker is introduced by Beth Biro, I wanted to briefly do a land acknowledgement. Tyson Research Center is located on land stolen from Indigenous peoples who were forced west and south into reservations during the Indian Removal Act of 1830. And these nations include the Kickapoo, Osage, Kaskaskia, and I'm embarrassed to say I don't yet know how to pronounce this fourth nation. And that this is a journey for us in learning the details of the history of the site. We want to get it right. So if any of you out there have information, are local and know some of the history of Indigenous peoples in our area and specifically at Tyson, we'd be happy to get that information from you. But we want to do this land acknowledgement, not because it is the cool thing to do these days, but because it aligns with our values and certainly because we are deeply involved in the land there through the research that we do, which oftentimes involves questions that are to understand what happens at the human environment interface. I also want to acknowledge that it's Pride Month. So we're very grateful for a very diverse group of folks in our community and globally, and we'll celebrate Pride throughout this month. With that, I'm going to hand this over to Beth Biro, Tyson staff scientist, to introduce our speaker today.
Thanks, Kim, for that wonderful welcome and acknowledgement. I would also like to thank all of you for tuning into the first Tyson Summer Seminar Series of 2021. We are so excited to have you all here, and we're extremely excited to welcome Dr. Nick Barber back to Tyson, who has come back to Tyson to talk about his work on Tallgrass Prairie restoration. But first, I wanted to tell you a little bit about Nick. I first met Nick in 2007. We were both at UMSL together. He was working on his PhD at the time, and I was finishing up my master's at Tyson. Nick quickly became one of my fellow ecology nerds, and he would always share all of his bird sightings on the Tyson Wildlife Board we had at the time, which we need to bring back, I think. We loved having Nick in the Tyson community. He was always willing to volunteer on some of our all hands on deck projects, which one may or may not have resulted in taking a trip to the hospital to get stitches in the head. Nick really took one for the team there. Nick's PhD work at Tyson included building a lot of bird exclosures out of netting. He started in the winter to be ready for the spring. He moved all his operation in the big Tyson classroom at the time so we could all work together on our various projects. Many a times Nick would be sewing a net together and one of the Tyson dogs would fall asleep in the middle of it and we'd have to free him from it, and it would always result in hilarity. After completing his doctoral degree at UMSL, Nick went on to do his postdoctoral work at University of Massachusetts Amherst with Dr. Lynn Adler, followed by an assistant professorship at Northern Illinois University, where he stayed on for 6 years before moving on to San Diego State University in 2018. He's been continuing some of his research from Illinois and expanding his research in his San Diego lab. So last year, Nick was awarded 2 large NSF grants, first with collaborators from Morton Arboretum, San Diego State University, Northern Illinois University, Middle Tennessee State University, and the Lawrence Berkeley National Laboratory, studying how plants shape the structure and function of soil microbial communities. And the second big award he was awarded will study how trait-based plant community assembly and grazing affects ecosystem function in tallgrass prairies. And that's with his collaborator, Dr. Holly Jones, who gave a wonderful Tyson seminar back in 2018 for those of you who are here to remember. So Nick has always been a very gracious and helpful person. And with all of his successful collaborative projects, it looks like he's merged his personality into his science. And I know he has so much to offer the scientific world. So I'm so excited to hear more about his research and where his group is going. So please help me welcome Nick to the Tyson Seminar Series.
Thanks so much, Beth. Hope everyone can see me, can hear everything. Let me know if there's any problems. Send me a message. I really appreciate getting to come here and give this talk. it's really fun. I have a lot of fond memories of Tyson, and I'm hoping I can get back and see it in person someday. But I'm hoping to give a little background information about the research I've been doing for the past few years, starting in Illinois and then continuing some of this work here at San Diego. One of the things I've been trying to do, though, is I know we've spent a lot of time on Zoom over the past year plus, is to try and make these Zoom seminars a little more interesting. I wanted to try something a little bit more interactive. If I can get this set up. So if you have a phone with you, hopefully you probably do. If you go to slido.com, maybe you've used this before, and enter slido.com and enter this code, you should see a question pop up. You should see a question about your study organism. I know we have a lot of people joining us, including students who maybe are just starting in research, or folks in the community who may not be scientists. So if it isn't a study organism you study, maybe something just really interested in. But if you enter that code 6728, I'm doing along with you, you should see a question pop up. And you should be able to enter something you study. I'm just going to type in plants real quick. And if you do that too, we should start to see study organisms pop up. And I like this as a way to understand the audience I'm talking to and see the diversity of organisms. A lot of folks studying trees right away, I see. We'll take a minute to do that while I just kind of touch base and cover what Beth was just talking about, a little about my background, introduce myself. So like Beth mentioned, I actually started in St. Louis. I went to SLU for my undergrad. And after 4 years at SLU, I moved across town to UMSL. And looking then for a field research site for my dissertation work, I ended up out at Tyson. I carried out my research there studying trophic cascades, looking at insect herbivore communities on oaks. I actually started work in the oak canopies using this big bucket truck that did not go real well. So I mostly kept my feet on the ground after that. And actually, I wrote a lot of my dissertation in the headquarters building at Tyson, looking out, watching the LLC being built. So I have a lot of fond memories of my time there, especially the community of people there. It's been really fun to see people I met as students, as summer interns, go on to great careers in science, and now get to see them at conferences and follow them on Twitter and read their papers and so forth. So that's been a really a fun thing. So I see a lot of, we can look at the word cloud now if you can see it yourself, a lot of folks studying trees, plants, ticks, and bird-toed ticks. Yeah, there's definitely, that's good memories of ticks at Tyson. But I see bees, I see people, songbirds, invasive plants, mammals, lizards, bacteria, tiger mosquito, really wide variety, spiders, fungi, a wide variety of organisms, which is fun to me to see that. And it really maps onto my own research over the years, has covered a lot of organisms too, from plants to insects, fungi, bacteria, and now even looking at large, very large mammals. So after I finished my dissertation, like Beth said, I went up to UMass to carry out my postdoctoral work, looking at above below ground interactions in agriculture, and then started my first faculty position at Northern Illinois University, where I spent 7 years and began the work I'll be talking about today. And the last 3 years, then I've been down in San Diego State. When I arrived at NIU, when I got out there and saw the area, this is west of Chicago. As any of you who have driven from St. Louis up to Chicago know, as you drive across Illinois, most of what you see is something like this, at least in the summertime, or its equivalent of soybeans. This is the eastern end of the corn soy ecosystem, one of the largest contiguous habitat types that exists in the world, stretching across much of the central US. So this is what I saw around Northern Illinois when I arrived there. But of course, if I were there 200 years earlier, I wouldn't see a tall cornfield like this. I'd probably see something more like this traditional tallgrass prairie, probably not even this many trees. The tallgrass prairie was this enormous and diverse ecosystem that once stretched across the center of North America from what's now southern Canada down through Texas, in this broad band, extending then east through Iowa and Illinois and adjacent states, with even possibly pockets as far east as Ohio or Alabama, depending how you define your prairies. It's a beautiful and biodiverse habitat type, but it's one of the most endangered and most degraded habitat types in the world too. The vast majority of the tallgrass prairie is lost or has been converted, especially to agriculture, all throughout the US. Here's one map representing where present day remnant prairies may still exist. In Illinois, the official number is over 99.99% of the original tallgrass prairie has been converted to agriculture or urban areas. And yet, even as those losses were taking place in the late 1800s and into the early 20th century, it was recognized by some ecologists. They were recognizing the prairie was disappearing and began asking questions about this habitat if it could be recreated. Although Leopold in the upper left was part of some of these early experiments in Wisconsin, sometimes working with civilian conservation corps crews to experiment with transplanting sod or planting seeds, or even early experiments with fire. And this laid a lot of the groundwork for modern prairie restoration practices, in which seeds are collected from existing restorations or remnants. They're spread over fields, often former ag fields during the winter, with prairie plants then germinating the following spring. But restoration work is an ongoing process. It requires active management. And in our world today, that often means regular application of herbicide to remove exotic weeds, and the application of prescribed fires. And this is really important, disturbance fires, because it suppresses many of those exotic weeds. It also suppresses woody shrubs and trees and maintains the prairie as a grassland. It's also important because it's a historical disturbance on these lands. Fires from lightning could burn over large areas. And importantly, although early restoration workers in the 20th century didn't recognize this, we acknowledge today that these fires can mimic those that were used by indigenous people on these lands for thousands of years through their own stewardship to maintain this diverse habitat and encourage grazing. The site that I work at is the homeland of several groups, including Peoria, Potawatomi, Oceti-Sakowin, Miami, Kickapoo, Sauk, and Meskwaki people. And their absence and the absence of their stewardship had profound impacts on prairies. These are some aerial photographs of an area just next to one of my study sites, showing how the absence and the suppression of fires transforms open prairie and savanna habitats, the bottom half of the left photo, into much more closed forest habitats today. And so prescribed fire is a really necessary tool for prairie restoration to maintain these prairie habitats and a lot of the oak habitats that we see down through Tyson as well. There's another aspect of prairie restoration that's connected to management, and that's the fact that restored prairies tend to lose plant richness and diversity over time. This is a really common phenomenon. Here's 5 different graphs showing examples of studies showing declines in richness or diversity as restorations get older. Often this is due to losses of forbs as grasses, especially warm season C4 grasses, can spread and outcompete them. And this actually has been part of the motivation for the reintroduction of American bison and potentially other grazers in some restored prairie projects across the Midwest. Bison often prefer to feed on grasses, and so their grazing may suppress those grasses, those competitively dominant grasses, and allow other plant species to continue existing in restorations. So these 2 disturbances, fire and grazing, are really important historically, and they're important today in shaping grassland communities and ecosystems. Okay, so let me have another quick question here. Let me pull it up and launch it. So when you think about restoration, I like to ask this question to different groups I'm talking to. When you hear me talk about restoration, what is it you think of as the most important goal? Is it the goal to maximize biodiversity, to recreate something historical, to match a current reference site, or to maintain ecosystem functions or the services that benefit us? Or do you think of it as focused on particular species of conservation concern? And I know that maybe more than one of these might be really a goal you think of, but if you had to choose, what do you think is the primary goal? Let me see if the pops up on here. Curious to see. And so we see so far, most people are saying maintaining ecosystem functions and services as a primary goal. After that, maximizing biodiversity. And fewer people saying matching reference sites or protecting particular species of concern. And I'm going to try and touch on some of these ideas throughout the talk, because often we have multiple goals, right? And one of these goals will support the other. But it's curious as I talk to student groups or talk to restoration practitioners or other scientists, we often think about different goals or different purposes, different motivations for restoration work. And I see ecosystem functions and services jumping up to the top, as is often the case when I talk to practicing scientists. So along with these different goals, one of some kind of common things we see in prairie restoration is that the restoration, management, ongoing monitoring often has a focus primarily on plants for understandable reasons. Establishing a plant community is part of establishing the grassland habitat. There's also often an assumption then that by supporting plant diversity and maintaining high plant diversity, we'll also be able to maintain high animal diversity. The plant diversity will drive animal diversity. And although it's obviously a concern for a lot of us, a lot historically has been kind of may overlook ecosystem functions at the expense of a focus on biodiversity, and particularly plant biodiversity. So I try to address these points in my research in asking how prairie restoration and ongoing management of restorations can shape these communities and ecosystems. In my talk today, I'll touch on 3 different areas. I'll talk for a little bit about animal communities and animal diversity, a little bit on soil communities that we've done, and then I want to mention a little bit about these 2 new projects that Beth brought up that we're just starting that grew out of a lot of this work I'll talk about today. So I work at, most of my work is at a site called Nachusa Grasslands. This is owned and operated by the Nature Conservancy. It's a couple hours west of Chicago. It's a large area. It's over 1,500 hectares now of remnant and restored prairie, as well as savanna habitat and some wetlands as well. In this area, at the end of the last ice age, the glacial wash kind of washed across west from the Chicago region and washed away a lot of the topsoil in certain areas, leaving these kind of rocky outcroppings that couldn't be plowed, couldn't be turned into farm fields. And so these are a lot of the remnant prairies that remain in the region, these kind of rocky hilltops that may have been lightly grazed, but a lot of the plant community is still there. And so some of these were protected, and beginning in the 1980s, the low-lying areas in between these rocky outcroppings that had been used for agriculture, restoration often started to focus on these areas and replant prairie and reconnect some of these hilltop remnants. The Nature Conservancy at this site has continued to plant new sites annually as they acquire land. And importantly for us as scientists, they use really consistent approaches in their restoration and management with a frequent use of prescribed fire and careful weed control. Also excitingly, in 2014, they introduced a herd of American bison that have free range over about half the preserve. And so they can impose their various impacts through grazing and wallowing and waste deposition and trampling across the habitat and shape it in new ways. So the work I'll show today grew out of a project that I developed with Dr. Holly Jones, who's an important collaborator of mine. And we call this the Refuge Project for Restoring Functioning Grassland Ecosystems. We've been focusing on 13 focal restorations that were planted over a range of years. Some have bison, some don't have bison access. We compare bison impacts. We also look at times at 2 of the large remnants. Most of the remnants are quite small. These ones are large enough that we can look at organisms in them. And most of these sites, like I said, they're burned off on average about every other year, although it's not a perfect schedule. So with this setup, we're able to study these replicate prairies that vary in age, bison presence, fire schedule. And we were fortunate to get an NSF EAGER grant to start this and study a variety of plants and animals and how they interact. I'm going to focus today for a little bit on I'll start talking about 2 of these animal groups that I hadn't worked with either of these before, but I started working on ground beetles and dung beetles and prairies. I found them to be surprisingly charismatic animals. Ground beetles are a really diverse family, about 40,000 species worldwide, several thousand in North America. They're in a range of different habitats. They can be really important predators of arthropods and seeds, both in grasslands and in agriculture. In agriculture, they're potential biocontrol agents when they eat weed and insect pests. And dung beetles include species in a couple of different families and several subfamilies. They, of course, use animal waste as a food. The work that I'm going to show here is from master's thesis work by 2 of my former students, Melissa Nelson and Cheryl Hassler. Melissa focused on ground beetles, and Cheryl, who's now a PhD student at UIC, worked on dung beetles. And in both cases, they census communities at this site using pitfall traps, either passive or baited with fresh bison dung, which we had endless access to. And so they asked how beetle communities change as restorations age, when fire is applied, whether or not bison have access. And they answer these questions both by looking at traditional measures of abundance and species richness and diversity, but also looking at the functional traits of these organisms, the characteristics that determine their distribution, their demography, their ecological roles. For beetles, this included things like size. You can see the size variation in the middle here of ground beetles on the left and dung beetles on the right. Their diet preferences, whether their beetles are carnivorous or omnivorous. Their mobility, some of the ground beetles are flightless, others are strong flyers. The time of day or time of year they're active, and behaviors as well. Most the dung beetles have a variety of behaviors. I know we often think of these dung rollers that make a ball of dung and roll it away. Those are actually pretty rare at Nachusa. Most of our dung beetles are either tunnelers that dig up to the dung from below ground and pull it down to chambers, or they're these dwellers that just dive in head first and they just live their best life in a big pile of poop. So we can see how these different traits, these particular traits, relate to different characteristics of the environment for both of these ecologically important insect groups. Melissa collected over 3,600 ground beetles from 41 species when she did her work in 2018, and found that the age of the restoration was most important in explaining variation in the communities, that as these restorations get older, we find higher abundance and lower richness of ground beetles. So in these figures, the x-axis is the age of the restoration from young to old, going left to right. And the triangles are just the two remnant sites just for reference. They don't really have an age. But as you can see, as these sites get older, more beetles are present, but from a smaller number of species. When we look at their traits, the functional trait diversity, this is functional dispersion, one way to measure the variation in traits of those beetles, we find this kind of a convergence on beetles with a rather limited set of traits. They tend to be species that are large, occur late in the season, are flightless, nocturnal, and are carnivorous. And these three species on the right characterize either all or some of these traits. So we have this convergence on a kind of limited ecological set of species with this one particular trait syndrome. Shifting to dung beetles, Cheryl collected over 27,000 dung beetles in 2 seasons. In this case, not surprisingly, bison presence was an important determinant of the dung beetle community, that when bison are present, dung beetles are more abundant, and there's also higher species richness. There were relatively few effects on dung beetle trait patterns, although their dung beetles are somewhat smaller in older prairies. When we look at the different measures of their average mass, these are community weighted mean measures of size. Although bison might actually kind of mitigate this and support larger beetles in these older sites, which may be important, you'll see in a second. So that tells us a little how the community is changing over time, or with whether or not bison are present. The other question Melissa and Cheryl asked is what these changes mean for the functions that these beetles are carrying out. In one case, seed and arthropod predation, which Melissa measured, putting seeds or waxworm larvae in little petri dishes and set them out in cages to measure predation rates. Or dung decomposition. Cheryl collected enormous quantities of dung that we had in our lab for a while. It froze in, and then we thawed it, homogenized it, and she created these artificial dung paths to measure how it decomposed with time. So first, looking at predation, seed predation and larvae predation were both on average reduced after prescribed fire. There was less predation of seeds and larvae in years where prescribed fire had been applied before the growing season to these prairies. Seed predation was mostly unrelated to the beetle community. One possibility is that ants really might be driving this pattern rather than these beetles. But arthropod predation really did correlate with the ground beetles. In particular, correlated strongly with carnivore activity density, the carnivore abundance. The larger the number of carnivorous beetles present, the higher the rates of larvae we saw eaten. And along with that, after prescribed fire took place, there was a reduction in these beetles. They were significantly less abundant. So fire really does seem to be shaping this community in this way that changes predation rates on other arthropods. We also saw some fire effect on dung decomposition. There was a trend for reduced dung decomposition after fire, reduction of this particular function. And also on average, there was greater dung loss where the average beetle size was larger, which matches work in a lot of other systems, that larger beetles even per their body size, per mass, are more efficient at removing dung and burying it and contributing to this decomposition. I'll emphasize, though, that dung decomposition was really difficult to measure with precision. Cheryl put a lot of work into this. It's just a challenging thing to measure this in the variableness of the real world. So kind of summarizing this then, for ground beetles, at least as restorations got older, communities consisted of fewer species that were mostly this syndrome of large, flightless, and carnivorous. And prescribed fire seems to reduce carnivores and then their predation on other arthropods. For dung beetles, we're not going to win a Nobel Prize for this. If you add large pooping animals to a prairie, it supports dung beetle communities. But it may even support larger beetles that on average are that can help promote dung decomposition. And on the other hand, prescribed fire might slow some of that rate. So one takeaway from this is that different animal groups respond to restoration management in different ways. But these aren't the only animals on the prairie, of course. We were part of this large group of folks at NIU that were studying other aspects of the community, including both plants and animals. Holly and her students have been studying small mammal communities. Rich King and his lab have been surveying snakes at Nachusa. And so we had this large multi-taxa data set that Pete Guyden in the upper left there took on as a postdoc and worked on to try and answer some other questions about these prairies. If I'll turn back to one of those assumptions we talked about earlier, the assumption that about plant diversity influencing animal diversity, we sometimes talk in restoration ecology about this field of dreams hypothesis named for the movie. One interpretation of this hypothesis is the idea that higher plant diversity directly drives higher animal diversity. And so one way of thinking that is that management actions that influence plant biodiversity, then those plant biodiversity will then drive animal biodiversity. So this plant's kind of moderating this effect of management on animals through this pathway we're referring to as plant-driven effects. Alternatively, or even at the same time, management, of course, can have direct impacts on animal communities, which we've been calling these management-driven effects. And so Pete wanted to ask how this variation we see in management, variation in the presence of bison, the age of restorations, and he also then coded out fire by how long it had been, how many months since the last fire took place, how these affect animal diversity, and whether that's occurring through plants or directly. Biodiversity is a complex thing, right? There's it's multidimensional. I've talked before so far. I would think about traditional taxonomic diversity, number of species, and the relative abundance. But we can also talk about functional diversity, variation in the traits of those species, or phylogenetic diversity, the variation in a range of evolutionary histories encompassed by a community. So Pete wanted to ask how these affected these multidimensional aspects of biodiversity. I'll present, I'm going to skip over functional animal diversity today just because it kind of mapped very closely onto phylogenetic diversity. But he looked at this and asked which of these effects plant-driven management might be stronger, or what might be explaining animal diversity for these four animal groups I mentioned, both ground and dung beetles and small mammals and snakes. I won't go into lots of detail, but Pete used piecewise structural equation models to kind of evaluate the evidence for these two groups of pathways, the plant-driven effects shown by the green arrows and the management-driven direct effects potentially shown by the blue arrows. And one thing this allows them to do is to compare their relative strength to see if one type of effect, plant or management-driven, explains more of the variation in animal diversity. So I think this is my last Slido interactive slide. Let me launch it real quick. So what do you expect to happen? Think about which do you think has a stronger impact on animal diversity? Is it this pathway through plant-driven effects or this pathway through management-driven effects? Do you think they have really similar effects, or maybe one just a little stronger than the other? So if you haven't read the paper, you can go ahead and choose which one you think, which you imagine has a stronger impact on animal diversity. See some answers rolling in. Everybody a second to think over that. And so as people answer, I see a few answers come in. It seems like people are suspecting maybe, or they're changing very rapidly now as more people answer. People seem to be guessing either that management effects are a little bit stronger, or that management and plant effects are similar from each other. So let's see. I'm going to apologize for the next 2 slides. There just aren't real easy ways to depict these figures given how many variables we include. But what you're seeing here is the arrows are indicating the significant pathways. The thickness of the arrow is a measure of their relative strength, how much of the variation they explain, averaged across multiple models. So here we're looking at effects on animal taxonomic diversity. The little icons represent the results for different animal groups for which that path was significant, with black meaning circles meaning a positive effect and red meaning a negative effect on diversity. I know it's hard to read these and go out, but the takeaway of this is first that each animal group responded differently. We see really different patterns, like we saw with the ground and dung beetles. Different animal groups are responding to management in very different ways and responding to plant diversity in different ways. We also saw that plant-driven effects, when they do occur, were primarily due to succession, due to age, not due to bison or fire effects on plant diversity. And overall, that management-driven effects through age and fire in particular were on average quite strong. So that's what we see for taxonomic diversity. Phylogenetic diversity we're similar, or even maybe a little more simplified, with weaker plant-driven effects and a really strong direct management-driven effect of age. So turning to our question then about which are stronger, you can probably see from these that the management-driven pathways, these management-driven effects are on average 6 times stronger than plant-driven pathways. This magnitude of effect is this absolute value of those path coefficients. So it seems that this direct effect of management is more important in determining animal diversity than the kind of indirect effect through plant diversity. So I want to mention a couple of things about this. Now, this does not mean that plant diversity is unimportant. That certainly is not the case. That obviously that's still an important goal. And in particular, there are situations for particular taxa, places where plant diversity is often positively correlated with animal diversity, but isn't always. You can't just assume that's going to be the case for all animals. What this does mean is that management actions are really important determinants of animal diversity. And so management choices should consider the impacts on animal communities in addition to the effects of those choices on plant communities, which I realize are often the main motivation for those actions. So I'll shift now to another aspect of prairies. Prairies can be really beautiful habitats, often because of this floral diversity that we often appreciate. That's great for supporting bee diversity. But there's a whole other side of prairies below our feet, right? In the soil environment. Prairie plants, as many as you probably know, can have really, really deep root systems, lots of their biomass occurring underground. That's why prairies can be so efficient, so great at storing, sequestering carbon. And those soil environments contain just an almost unfathomable diversity of microorganisms. And these microbes drive a lot of those critical ecosystem processes. They affect carbon cycling, they affect nutrient cycling, really critical parts of these habitats that they're not always considered or historically weren't focused on. Oh, and there's a picture below ground. So also, as time goes on after restoration, not surprisingly, when you shift from this really high intense agricultural soil management to restorations, we see a lot of changes. Very broadly, there tends to be increases in soil organic matter, increases in soil carbon, soil bulk density. These graphs on the right are from a couple different studies. Actually, I think the top is from a paper by Sarah Baer, and I think I first saw this data presented at a Tyson seminar back in 2007 or 2008. And you can see as restorations get older from left to right, you see an increase in soil carbon, contrasting with the very low levels in ag fields in the far left of the figure, and the high levels often present in remnant prairie soils. We also often see an initial rapid decrease in soil nitrogen because these soils aren't being loaded with fertilizers frequently. And then as organic matter and so forth builds, you'll see a slow increase in nitrogen later on. So we can assume then that if there's these large changes in the soil environment, it's likely to affect the organisms that live in those environments too. So I began collaborating a number of years back with Dr. Wes Swingley, who was another professor starting at NIU around the same time as me. And we started studying the bacterial communities in the soils at Nachusa using 16S RNA gene sequencing. We found a couple patterns in our initial work. One is that if we look at this, now looking just at bacterial classes, the diversity and the richness of classes tends to decline as these restorations age. They tend to be quite high in agricultural fields, the open symbols on the left, and they tend to be quite low in the remnants shown by the triangles. They decline over time. So on the one hand, these restorations are coming to look more like the remnants in this decline of richness and diversity. We also looked at the composition in a little more detail. We've repeatedly sampled a subset of restorations and a remnant in an agricultural field across the whole growing season from April to September. The figure you're seeing here is a non-metric multidimensional scaling ordination. The dots, the color represents which field the sample came from. The numbers are just what number sample it was. But if you haven't seen an ordination like this before, two dots that are very close together have very similar combination of a community. Dots that are far apart are very different. And so you see that the youngest restorations, the 1, 2, 3-year-old in blue, tend to cluster on one side of the figure. And as restorations get older in the pink and the red, they're converging with the remnant communities in maroon. So these, not only these restorations coming to resemble the remnants in richness and diversity, the composition, which classes we see in the relative abundances, look a lot like the remnants too, as these sites get older. Also interestingly, the farm field, the agricultural field is kind of an intermediate, is a mix both of these old and young prairies, which may contribute to that, that higher diversity and richness of their communities that we saw. And also excitingly, when we look actually at which groups are affected, some of the classes considered typical of remnant prairies, Verruca microbia, some of the acidobacteria that have been found to have relatively high relative abundance in remnant prairies were also becoming more abundant over time, suggesting that this process of restoration is returning these soil communities to something resembling what we see in the remnants. And if that's a goal, if that reference is a goal for it, this may be a positive sign of a successful restoration. So I'll shift now to talk about another, I should mention too, that we've been sampling this from 2013 to the present. We have over 350 soil microbiomes that we're really diving into and hoping to be able to report back on later this year, to look at this both in more detail, to identify which taxa really are driving these changes and potentially see bison impacts as well. Those results, though, and this continued sampling we've been doing really made us think a lot about what is driving these changes. We know the plant community and restoration obviously is changing over time too. We know that plant communities can have strong effects and strong impacts shaping communities of soil bacteria and archaea and fungi. One of the possible mechanisms for these effects are root exudates, these organic compounds that are released from plant roots into the surrounding soil. A really large, significant amount of carbon captured by plants in photosynthesis is lost or is expelled from roots in a really wide variety of organic molecules. And those compounds then can be resources for soil microbes. So variation in root exudates due to variation in a plant community might help to explain the structure of soil microbial communities and potentially those important functions those microbes carry out in soils. So as Beth mentioned earlier, last year, a group of us from several different institutions were fortunate to receive an NSF Rules of Life grant to study these ideas of how plant communities shape soil microbial structure and function via root exudates. And this has several components I just want to touch on in my last little bit of time here. One of the things we're going to do is characterize the root exudates of 127 different native prairie plant species. This is work I'm going to be carrying out with Andrew Hipp. It allows us to ask questions about those patterns of root exudates, phylogenetic questions like, do exudate blends tend to be similar when we're looking at related plant species in the same clade? We can also look at how they relate to traits. Are there particular plant traits, especially root traits, that can help explain differences in these exudate blends? Collaborators at the Arboretum, Megan Midgley, along with Kate Jalnina and Trent Northern at the Berkeley lab, are then going to look at how those exudates may directly affect communities. They're going to do these these soil incubations that they inject the soils with specific exudate blends from our plants and look at how the soil microbes change, how the community changes, to ask, do these microbes adapt to metabolize local exudates in their environment? Are there particular exudates that are utilized by given microbes using isolates they have? And then from these very controlled experiments, we're going to move out to a experiment at the Morton Arboretum. I know some folks at Tyson are familiar with this amazing experiment that Andrew and Megan and others at the Arboretum set up, in which they've assembled prairie plant plots that include both these 127 species growing in monoculture, and then also community plots with 15 species varying in their functional trait diversity and their phylogenetic diversity. And so we can look at the actual microbial community structure and look at their functions with metagenomic sequencing in both those monocultures and the experimental plots, and see, do exudates explain the actual monoculture soil communities that we see? And does that vary within or among clades? Does the plant community, its own phylogenetic diversity, drive microbial diversity in different ways? So Wes, who's a soil microbiologist and a microbial ecologist, is going to be helping with a lot of this. And then lastly, we're going to try and take this out really into the real world and build and test these predictive models of soil communities. Josh Phillips is a computer scientist at Middle Tennessee who builds these artificial neural networks. And we're going to use this to try and predictive models of structure and function, and actually test these models using our field data from Nachusa, where we have long-term plant monitoring plots, to see if can we predict the soil communities in those real prairies. And importantly too, can our models that are trained on one set of species predict the effects of other species, because we don't have, there's more than 127 plants at Nachusa. And that can be important to understand if we can then generalize some of these patterns to other grasslands and other habitat types too. So obviously this is a really large project we're going to be working on the coming year. I'm excited that our very first data is starting to roll in after kind of a slow start in the past year. We'll be doing some large scale sampling at the experiment this coming in July. The other question the other thing I'll touch on just to finish up here, when I think back to that Slido question I gave about goals of restoration, one of the possible goals I mentioned is this idea of supporting ecosystem functions and services. That was, I think, the majority of people who answered identified that as perhaps a primary goal of restoration. Thinking about ecosystem function, there's this long history of biodiversity, ecosystem function experiments in ecology, often manipulating the number of species in a community and demonstrating that in lots of cases, more species results in higher levels of various functions. Holly and I have been thinking about this and based on our pilot data at Nachusa and how we can kind of investigate this. Two of the critiques of these sorts of experiments that are sometimes made, critiques of these biodiversity ecosystem function experiments, are first that the experiment's diversity gradients or range of species richness often doesn't really resemble actual community assembly as it takes place in the real world. They're often made of randomly assembled groups of species, or they don't reflect the community turnover through which community assembly actually happens. Secondly, they often don't include trophic interactions. Those are some noteworthy experiments that have. So we're trying to address both of these points in a new VEF experiment that we're going to be setting up actually a week from Monday. I'm going to be up in Illinois planting about 8,000 prairie plants to address this. But the way we designed this experiment to address these concerns, first is that we're setting up these plots of prairie plants that vary in their number of species, but arrive at that number and arrive at the combination of species through simulated losses and gains that are non-random with respect to functional trait diversity. So in each of the cases, what we're going to do is start with a pool of a dozen species, shown by this black dot here, and then create plots through the removal of species from the species pool that result either in increases or decreases in functional diversity, and then add in species non-randomly, again, either increasing or decreasing functional diversity. And we can carry out all these different possible scenarios, different ways that species losses and gains through community turnover may increase or decrease the functional trait diversity of these communities, considering both above ground and below ground traits. And what this will essentially do then is we'll arrive at a factorially cross design that crossing species richness and functional diversity changes, but arriving there in these different ways. And we'll measure a range of ecosystem functions above and below ground biomass, nitrogen uptake, other functions that, and we can then use the price equation to partition changes in ecosystem function to understand how much of this change, a change in function, is attributed to changes in richness versus changes in assembly. So changes due to species losses or gains, or changes in functions due to the contributions of the function by the species remaining in the community, the part of the community that isn't changing in that turnover. And then lastly, to build in the trophic interactions I mentioned, half of these plots will receive simulated grazing, in which will remove biomass, plant biomass, into proportion to what the bison actually eat at Nachusa, which we've been able to determine using stable isotope mixing models based on stable isotopes from bison hair that we've collected. So again, we're going to be collecting a lot of data in the coming years with the goal of trying to understand these processes. We're setting up very soon. It's gonna be a busy week setting the experiment up. I also want to just mention that we're going to be hiring a postdoc later this year to work on this project who will be based at NIU. So anyone in the audience who is looking for that at that point in their careers or knows someone who is, check it out, keep an eye out. We'll have an advertisement for that hopefully by the end of the year. Right, last I want to acknowledge some folks who really support this work. Obviously my collaborators, Holly and Wes and Pete and Rich, and especially to a lot of the students, our students in our labs. We often have essentially co-advising students over the years, especially Melissa and Cheryl, whose work I talked about today, and other students from our labs have really contributed a lot of the data that went into the research I talked about. I also really want to importantly mention the Nature Conservancy, who's been an incredibly great partner for our research. The staff at Nachusa, Bill Kleiman, Cody Considine, and Elizabeth Bach make this work possible. They really give us a lot more freedom and a lot to work in these restorations. That isn't always the case at a lot of field sites where conservation really is the primary goal, but they have their mission includes serving and supporting science, which helps us a lot. The Friends of Nachusa is their nonprofit group that has supported a lot of our students with small grants and supports the volunteers and the stewards who do lots of the work at Nachusa and make it such a great place to work. I also acknowledge the range of funding we've had for these projects that's made it possible. All right, so thank you so much. I would love to hear some questions from folks and hope you enjoyed the talk.
Thank you so much, Nick. That was wonderful. If you were in person, you'd be hearing thunderous applause. We have a couple.
Thank you.
We have some people here in the LLC listening on the big screen, so thank you so much. So I just want to remind everyone that you can put your questions in either the YouTube chat, or it looks like we have a couple from the Slido website that Nick, that we did our polls on. So you can throw your questions in there, and I think Nick can have that too to see. So just going through the YouTube real quick, we have, a lot of students asking questions, and one of our undergraduate students asks, what defines a true prairie? I think that some other students have, kind of interjected with simple answers, but I think it might be a little bit more complicated, and maybe you can kind of discuss it a little bit.
Yeah, that's a great question. And I referred to those maps I showed that, sometimes I show pockets of prairies further east, and there's sometimes debates about what is really a prairie or not. And I think these are difficult things to decide. I know people have tried to define them. The other issue arises too is, I know we have a lot of oak savannas through parts of the Midwest. When does a prairie become a savanna? When does a savanna become a forest? And people tried categorizing those by different levels of canopy cover as well. But we often have prairies with scattered trees that may be more savanna-like. I don't have a great answer for that. I think when people try and define grasslands in general, it's a relatively low growing community, has a high abundance and high cover of grasses and a dominance of non-woody plants as opposed to woody plant structure. But it's a good question. And this comes up too when we talk about what's a successful prairie restoration. Have you restored a prairie? A lot of times too, my opinion is to think of the alternative. If it's a completely unrestored site that's in has lots of exotic weeds in it versus something that might have more native species and support a lot more higher biodiversity, may not be a perfect prairie, but that still has benefits for us in the world today.
Absolutely. Thank you for clarifying that. Let's see. So Jonathan Lalsos asked a question, and I think it refers to your second question in the Slido about, so what does using the term restoration in the question change the response? So I think everybody chose,
Oh,
do you got it?
pull that question up. Yeah. Can I see that question?
Let me see. I don't have the question anymore.
Oh yeah, what's the most important goal of restoration? I think maybe he means as opposed to the important goal of conservation in general, maybe is the way to think of that. Yeah, I mean it's kind of an unfair question. I think what I found is it's interesting talking to because there's multiple goals, and I think everyone who works in conservation recognizes that. I'm interested because I think a lot of times when I teach conservation biology, I find students are often think of it with this historical view. The goal is to recreate what happened in the past, which is a great conversation starter about whether we can recreate the past, given that we're in a different climate and we have perhaps different species. A lot of times too, the motivation for conservation may be protecting particular species of conservation concern. I can tell you that Nachusa's visitorship went up more than tenfold when they introduced bison. People aren't going out there to see ground beetles. They're going there to see the bison. And that can be an important motivation for restoration that still serves these other purposes too. But in terms of restoration itself, then yeah, I think then another way to think of this is that restorations are human created and human managed environments. And that may help justify why supporting ecosystem services that benefit humans is a totally justifiable and reasonable goal for that. And likely to achieve that, you're going to have to do things like significantly increase biodiversity, create something that may at least overlap somewhat with historical conditions, and probably support some species of conservation concern too.
Yeah, great, thank you. Let me check. We got a lot of questions rolling in now. Okay, let's see. I know James Traeger says hi, and he says, I may or may not ask you about ants. And there's an anonymous question in the Slido that is about ants, so I wonder if it is.
Oh, if it is, so last time I saw James was out at Nachusa, and I recall saying, James, you should totally come up here and study ants because nobody's studying ants, and we really need someone with that expertise. I know that Melissa frequently saw ants in there, whether they were exactly foraging on those seeds or not isn't clear. There's potentially other seed-eating organisms too that may have accessed her little cages too that may be contributing to that. But yeah, we don't really know in particular. It seems that at least the seed predation as we measured it doesn't seem to be strongly related to any of the characteristics of the ground beetle community as we measured them.
Okay. You're having lots of virtual applause through the chat. Student asked, does increasing or decreasing the functional diversity of plants affect the functional diversity of other taxa, such as beetles, or vice versa?
Let me try and jump back to, without jumping too many, I had to jump a few slides back. Quickly, I'm going to pull up the structural equation slide looking at, so functional diversity was, in this case, was fairly strongly correlated with phylogenetic diversity. There was, so there were not positive correlations we saw, at least this from a functional diversity of plants driving phylodiversity of beetles. We saw that instead for snakes and small mammals. There weren't real strong direct effects of that. Trying to think of other times we've done that analysis. It doesn't seem to be real strong. One of the things that often ends up driving the functional diversity or phylogenetic diversity for these plant communities is the relative grass abundance, relative grass dominance. That when a site is really grass dominated, it ends up having, by all measures of diversity, lower diversity. Those communities don't seem to necessarily be particularly detrimental for ground beetles. In fact, those carnivorous species of flightless carnivores seem to do just fine in those older grass dominated communities.
Okay, LLC postdoc Sasha Heath asks, beyond perhaps increased fire frequency, what expectations do you have for the effects of increasing temperatures and reduced precipitation on prairie restoration?
Repeat the what was the driving effects on prairie?
Hold on, I'm unmuted. Okay. Beyond perhaps increased fire frequency, what expectations do you have for the effects of increasing temperatures and reduced precipitation on prairie restoration?
So this is actually something I was talking with some folks earlier today. There's some fascinating work coming out of some groups looking at how precipitation in the first year of growth for a prairie affects the outcome of that prairie, and that in some cases a very rainy year can actually be bad for prairie outcomes because it allows exotic weeds to really take off and dominate and end up kind of suppressing a lot of the native prairie plants. Whereas a drought year, if those prairie plants can hang on and get a little bit established, because exotics are less likely to succeed, then that may end up being a more successful prairie in the long run. So there may be situations where some reduced precipitation, if it's a case where it reduces exotic species, might help restorations be more successful in some cases. There's going to be a lot of contingencies about that too, of course. It's going to depend a lot on input of exotic species as well, and whether that mechanism actually takes place. Where we go. Sorry, thank you slide. There we go.
Okay, Titan staff scientist Katie Westby asks, do you see similar changes in pollinator diversity and abundance as the prairie plants age that you do with the ground beetles?
And I can't remember results at the top of my head. So Beth Ann Berninga-Sokolar and Sean Griffin are both postdocs. Beth Ann is at University of Minnesota. Sean is at UT Austin, I believe. I would highly recommend either of them as seminar speakers in the future. They're both great scientists. They've been studying the bee communities in these same sites for a number of years. I try to remember off the top of my head some of their results. One of the important results they've found is that in terms of looking at the bee community in a restoration, the larger scale surrounding habitat can be an important part of that explanation, in particular adjacency to some savanna or forest habitat, because it may provide a different nesting habitat, and those bees are just coming out and foraging in the prairie. And so that kind of supports the idea of a mosaic of different habitat types around these restorations to support bee diversity. They found that it isn't super strongly coupled to flower diversity or flower abundance. It seems like it's more of this landscape level pattern that may explain bee communities. But check their papers out because it's more detailed than that, and they did really fabulous work.
Undergraduate student asks, how do dung beetles contribute to the ecosystem as compared to other organisms like fungi that break down waste?
Oh, that's a good question. One way to think of it is there's certainly a partnership there too, that by removing dung, putting it in the ground, whether through rolling or through burying it. Now, a lot of times they're using it to provision for their offspring are eating it, but putting that down in the soil can help facilitate those interactions with soil fungi and allow soil fungi to break down that dung as well. In terms of I don't know if I off the top of my head can come up with numbers of like, if a pile of dung is sitting on the soil surface, how much of that is being taken away by beetles versus fungi. I don't know if offhand, but there's interactions between them as well, of one facilitating the other. Another factor I'll mention is because most people are surprised by this, is that there's obviously a lot of nutrients in dung. As that dung sits out, even as it's being eaten or decomposed, there's still a lot of loss of some nutrients of nitrogen. Nitrogen can kind of volatilize as ammonia into the atmosphere, and so a pretty significant amount of nitrogen can just float away in the air rather than being returned to the soil from decomposition, which is one of the ways that long-term repeated grazing on a site without nitrogen inputs can lead to nitrogen deficiencies in soils in some parts of the world.
Wonderful. I think we have one more question from Slido, anonymous. If age is the biggest driver of animal taxonomic diversity, what does that mean about management actions? Does it just take a few years for species to immigrate?
So yeah, I think that, and this is one, I think maybe another important caveat of our study, is the range of management actions we looked at is what was available to us. So although we had a range so fire, time since fire varied from just a few months to a few years. That's still a rather short fire gradient compared to, say, the research at Kanza, where they have sites that have gone years, decades without being burned, have a much longer time since fire gradient. That may be one reason that the fire impacts weren't particularly real strong in shaping these communities. Essentially, all the communities we study are relatively high frequency fire communities, like a lot of prairies would have been historically. In terms of bison too, that's a great question. Is it going to take more time too for these bison impacts to arise? We've already seen some effects, like I mentioned. There's been changes in ground beetle community composition. I didn't show those data of the relative abundances of different species in response to bison. It looks like bison may even, with some very preliminary analysis, be shifting the soil microbial communities too. And they do seem to, in some of the sites, having this effect of suppressing grasses and increasing the cover of forbs. So that's just after a few years. We suspect that after, hopefully after a couple of decades of bison presence, we may see a stronger factor or impact of bison on both plants and animals. For now, I think that age is one thing. It's the longest gradient we have to look at, which may explain why we saw some of the strongest effects from that.
I'll give it a couple more seconds. If anybody has any last minute questions, please put them in the chat. There was one other comment on Slido from Anonymous, is commenting on the definition of the true prairie. That never been plowed is part of the definition of a real prairie.
Yeah, that's a great point about what makes a prairie. If it's had some time, even if there's some plants there, some people say it needs to be reconstructed or restored. And that's part of the value of, there's so few of these areas of these unplowed prairies across the Midwest that makes them so valuable. I also see a comment from James. James, I'll send you an email with some of the publications. Happy to help out with that.
Well, that's very nice of you, Nick. I think that is all for the questions. And I just really want to thank you again for an awesome talk. And thank you for being a wonderful first seminar this season. Thank you so much.
Thanks for having me today, Beth. I really appreciate it. Thanks everyone for coming in.